Controlling one of Australia's worst invasive species may involve genetically engineered cannibal toads.
So the whole reason these animals are such a problem is these poison glands you've got on the neck.
When we hear the name Peter Pan, we think of a boy who never grows up, who flies away from responsibility, and lives forever in a storybook world. But in Australia, there's another kind of Peter Pan, one bred in a lab, designed not to fly, but to stay in the water and eat its own kind. This is not fiction. It's a radical scientific response to one of the worst ecological disasters in modern history. A creature created to stay young, stay hungry, and suppress its species before it ever reaches land. But despite promising results, it has never been released into the wild. So start right now to uncover the reason why. The pond that stays forever. In stories, Peter Pan lives in Neverland. In science, Peter Pan lives in a shallow pond in northern Australia and never leaves. It has no wings, no fairy dust, and no future beyond the waterline. What it does have is an appetite for destruction, focused entirely on its own kind. This version of Peter Pan is not a character, but a cane-toad tadpole, genetically modified so it never grows legs, never crawls onto land, and never becomes an adult. It remains in its larval state for weeks longer than normal, feeding voraciously on cane-toad eggs. Not metaphorically, literally. Its design is built on a brutal biological truth. Young cane toads already practice cannibalism. These gene-edited ones just do it longer, better, and with lethal efficiency. The idea sounds almost poetic, a creature that never grows up, existing only to curb its own future. But it didn't begin with poetry. It began with a crisis. Because before Peter Pan, there were millions of other toads, older, stronger, and far more dangerous, spreading across the continent with no one to stop them. And to understand why this new solution was ever considered, we have to go back to the beginning, when Australia made one of its most costly ecological mistakes. An island that keeps forgetting. Australia is a land shaped by isolation and scarred by biological optimism. Time and again, well-intentioned interventions have backfired into ecological disasters. European rabbits, red foxes, and feral cats were all introduced with little understanding of the consequences. But none have left a legacy quite as toxic as the cane toad. The story begins in Queensland in the 1930s. Sugarcane farming was booming, but a tiny pest called the greyback cane beetle was eating away at profits. Desperate for a fix, Australian scientists turned to a toad from far away. Reports from Puerto Rico claimed this species devoured beetle larvae. But rather than run extensive trials or assess long-term risks, officials relied on anecdotal evidence and rushed to act. In 1935, just over 100 cane toads were imported from Hawaii, bred in captivity, and released near sugar plantations. The idea was simple: let the toads roam and they'd handle the beetles. But the beetles lived high up on the stalks, well out of reach. The toads never touched them. Instead, they found a feast in Australia's lowland ecosystems. With no natural predators and virtually limitless habitat, the cane toads didn't just survive, they spread relentlessly. And by the time anyone understood what was happening, it was already far too late to undo. The monster multiplies faster. Cane toads didn't just spread, they transformed. As they invaded northern Australia, their bodies began to change. Legs grew longer, muscles strengthened. Their livers, the site of toxin production, swelled in size. Within a few decades, they were traveling 5 to 10 times farther per year than their South American ancestors. Scientists now describe them as frontline invaders, organisms evolving in real time under pressure. What made them unstoppable wasn't just their mobility, but their chemistry. Cane toads carry bufotoxins, potent compounds that target the heart, nervous system, and blood vessels. In South America, predators evolved to avoid or resist these toxins. But in Australia, native species had no such defenses. The effects were immediate and devastating. Monitor lizard populations plummeted. Freshwater crocodiles died in large numbers after ingesting just one toad. Even brown snakes, among the most venomous reptiles on Earth, succumbed within minutes. Attempts to control the toads seemed almost futile. Community toad busts collected millions, but the population bounced back within weeks. Chemical deterrents failed. Biological controls raised unacceptable risks to native species. By the early 2000s, the toad's territory had expanded across more than 1 million square kilometers, an invasion powered by reproduction, adaptation, and an almost complete absence of natural checks. Faced with a creature that had rewritten its own biology, scientists realized they needed to target the only stage in its life cycle where it might still be vulnerable, the water-bound beginning. Predators, eggs, and cannibal codes. Even without genetic edits, cane toad tadpoles are already their own worst enemies. Unlike most amphibians, their young don't just avoid siblings, they hunt them. When cane toad eggs hatch, they release a chemical signal into the water. That scent draws older tadpoles in, triggering a frenzied feast. In the competitive stillness of a shallow pond, the most dangerous predator of a cane toad is another cane toad. This bizarre twist is what led scientists to a new idea. What if they could design a tadpole that stayed in this hungry state longer? A version that never reached land, never bred, and never stopped eating eggs. Using CRISPR, clustered regularly interspaced short palindromic repeats technology, a team led by Professor Rick Shine disrupted the genetic signal that tells a tadpole to grow up, specifically by disabling its thyroid hormone pathway. Without that hormone, the toad remains aquatic and juvenile. These lab-created Peter Pan tadpoles, as they came to be known, weren't just permanent children. They were engineered cannibals, slightly larger, stronger swimmers, and far more efficient egg consumers. In controlled experiments, they devoured up to 4 times more eggs per day than their unmodified peers, and they did it for weeks longer. But even more striking was this. When researchers modeled their impact, they realized that just a few dozen Peter Pans in a small pond could wipe out tens of thousands of eggs before a single hatchling emerged. Yet for all their potential, these creatures would never leave the water or reproduce. The danger in designing nature. Despite their effectiveness in the lab, Peter Pan tadpoles remain trapped in trial tanks. The reason is not failure, but caution. Because even in tightly controlled settings, nature finds ways to surprise us. Researchers observed that these modified tadpoles occasionally consume the eggs of native frogs, albeit at much lower rates. The behavior wasn't aggressive, it was incidental, but it was enough to raise alarms. A non-zero risk to native species means no open release. Every Peter Pan test, to this day, is restricted to enclosed water bodies, fenced reservoirs, livestock dams, or sealed-off ponds where scientists can intervene at any moment. Even before these trials, the team had already learned hard lessons. In early CRISPR experiments, they created albino cane toads by editing a gene linked to pigmentation. The results were supposed to be straightforward. Instead, the albino tadpoles behaved differently. Slower reactions, poorer vision, increased vulnerability. They even underwent metamorphosis earlier due to stress. One simple edit had triggered an unexpected cascade of physiological changes. That's the quiet danger of this work. CRISPR can be precise, but biology is not a closed system. Turning off one switch may dim lights in places researchers never intended. Regulators understand this, so even with no risk of reproduction, no chance of spreading, and no observed long-term harm, Peter Pan remains a hypothesis, still pond-bound, still under observation. Until risks can be fully ruled out, this radical solution must remain contained. Because the last time Australia released a species without asking enough questions, it took less than a century to lose control of a continent. The dry lines they cannot cross. Peter Pan is not the only idea to emerge from Australia's war against the cane toad, but it may be the most cautious. Others were more ambitious and more dangerous. In the 1990s, researchers considered using chytrid fungus, known for wiping out amphibians worldwide, to target cane toad eggs. But the risk to native frogs was immediate and catastrophic. That plan was abandoned before it began. RNA interference showed early lab success, silencing metamorphosis in tadpoles. But in the wild, it degraded too fast. Hormone blockers were another option, but they harmed native species at unacceptable rates. These early attempts all shared a flaw. They acted on the entire environment. There was no off switch. By contrast, today's solutions are quieter and grounded. Native meat ants, for example, are not being released but encouraged. They naturally attack young toads during their brief, vulnerable time on land. In some areas, up to 90% of toadlets die from ant encounters alone. Meanwhile, pheromone traps lure tadpoles into mass capture, and aversion training, like nausea-inducing toad sausages, teaches some predators, like crocodiles, to avoid toads altogether. But the most strategically elegant tool may be water itself. Cane toads need it to survive the dry season, and artificial livestock troughs have unintentionally become stepping stones for their migration. Scientists and landowners now work together to seal these points off, a method called the dry shield. By cutting off access, they break the toad's expansion route without harming other wildlife. These layered methods offer no silver bullet, but combined, they buy time and space for science to proceed carefully. The battle against cane toads isn't over, and Peter Pan may never leave the pond. But should it? Would you risk releasing a creature designed to erase its own kind? Share your thoughts below, and if this story made you think twice, subscribe for the next one.